everyone. In this lecture, we're going to study the topic chemistry in everyday life. Soaps, detergents, medicines, toothpaste, explosives, fuels, rocket propellants, all these are examples of chemicals. So this gives us an idea of how useful chemicals are in our everyday life. Whether they're used in medicines, they're used in food articles, in preservatives, in toothpaste. So chemistry has very important applications in everyday life, right? We always study very theoretical concepts in chemistry. We hardly ever care about how applications of chemistry, are applied in real life. So this chapter is all about that. In this unit, we shall learn the application of chemistry in 3 important areas. One will be medicines, the second is food materials, and third is cleansing agents. So let's move on. First, we'll study about drugs and their application. So drugs are chemicals of low molecular masses which interact with macromolecular targets and produce a biological response. So when this drug action is useful, it's called chemotherapy. The usage is called chemotherapy and the drug itself is called a medicine. Right. Now, classification of drugs. So basically, there are 4 major bases on which drugs can be classified. One is on the basis of pharmacological effect. The second is on the basis of drug action. The third is on the basis of chemical structure. And the fourth is on the basis of molecular targets. So let's study more about the classification and the different kinds of drugs. The first classification is on the basis of pharmacological effects. So basically, this classification is on the action, on the final action of what the drug is doing as a medicine, right? Like analgesics have pain-killing effect. Antiseptics kill or arrest the growth of microorganisms. So that is their medicinal usage, right? The second is on the basis of drug action. How does the drug participate in a biochemical process? How does it react? So that is another mode of classification. The third is on the basis of chemical structure. Often structural features determine the pharmacological action. So all this is very interrelated because the drug action depends on the specific structural properties that it has. So yeah, like in this figure we can see the general structure of sulfonamides. Fourth is on the basis of molecular targets. So drugs usually interact with biomolecules such as carbohydrates, lipids, proteins, and nucleic acids. We'll study about the properties of these biomolecules in detail in the chapter Biomolecules. So drugs usually interact with them, and similar action mechanism implies similar targets. So if we know the way a drug functions, we can find out the targets, and the targets will mostly be similar in structure and otherwise. So now we move on to drug-target interaction. Yeah, macromolecules of biological origin perform various functions in the body. For example, proteins, a type of proteins are enzymes, right? They are biological catalysts. They help catalyze the reactions taking place in the cell. Another kind of proteins are receptors. They are crucial for communication system in the body. Then there are nucleic acids which carry coded genetic information for the cell. They are the information house of the cell. The third are lipids and carbohydrates. They are structural parts of the cell membrane. They constitute a cell membrane, give it structure and stability. Drug action on enzymes. So to study drug action on enzymes, first we need to study the catalytic action of enzymes. Enzymes have active sites that hold the substrate molecule in a suitable position so that it can be attacked by the reagent actively. So as we can see, there's an active site in an enzyme in which the substrate attaches itself so that it can be attacked easily by the reagent, right? So then it obviously means that enzymes are very shape selective, because only a particular shape of a substrate can fix in a particular active site, which makes them selective in nature. So substrates bind to the active site of the enzyme through hydrogen bonding, ionic bonding, etc. Now the second function of enzymes is that they provide functional groups that will attack the substrate and carry out chemical reactions. So on the whole, enzymes catalyze biological reactions, which are very important for life and survival. So that was catalytic action of enzymes. Now, how do drugs and enzymes interact? Let's see. Drugs interact with enzymes in two ways. One is drugs compete with natural substrates for their attachment on the active sites of enzymes. So instead of a substrate attaching to the active site of the enzyme, sometimes a drug gets attached. So the substrate cannot attach. So that particular reaction cannot take place. So these drugs are called competitive inhibitors. So they are inhibiting the function of a particular substrate or a particular biological molecule because they are attaching themselves to the active site of the enzyme. This is one way how drugs and enzymes can interact. Let's look at the second way. Now, some drugs do not bind to the enzyme's active site. What they do is they bind to a different site of the enzyme, not the active site, and this site is called allosteric site. Now, this attachment to the allosteric site changes the shape of the active site, and we had seen that the active sites in an enzyme are shape selective. So now if the shape changes, now it can no longer attract the substrate that it used to attract as its functional property. So this is the second way how drug-enzyme interaction can take place. Now we come to the next topic, which is receptors as drug targets. So receptors are proteins which are very important for communication, right? Communication between cells, so to say. So receptors are proteins that are crucial to a body's communication process, right? The shape of the receptor site changes to accommodate the chemical messenger. So basically, receptor sites are present on the cell membrane. They are on the outside of the cell, and the receptor site have a particular shape to which a chemical messenger attaches itself or sends its signals. So what happens is a chemical messenger is carrying some coded information, which is used for the communication of a signal, let's say a nervous signal. So what happens is there are these cells on the surface of which the receptor sites are present. The receptors have binding sites. The chemical messengers, as we can see very clearly in the figure, the chemical messengers send the message, attach themselves to the binding site. They send the message is transferred through the receptor to the interior of the cell, and the chemical messenger moves on. So this is how the receptors function, right? Now, how can receptors be targeted by the drugs? So receptors show selectivity for one chemical messenger over another because their binding sites have different shape, structure, and amino acid composition. Drugs that bind to the receptor site and inhibit its natural action are called antagonists. So now if in the receptor site, if a drug binds itself, then the chemical messenger cannot send its signals. So it is inhibiting the natural function of the receptor, and such drugs are called antagonists. Now, drugs that mimic the natural messenger by switching on the receptor are called agonists. Now, sometimes what happens is maybe a particular action needs to be performed, but the chemical messenger for that action are not present. So, this is an artificial way by which we can make the body function properly. Now, there might be drugs which are not the natural messengers, but they act as natural messengers because they know how to attach to the specific receptor site and send the signal. These are called agonists. So right now, so that was drug action on receptors and on enzymes, which are two important classes of proteins. Now we come to therapeutic action of different classes of drugs. Okay, so therapeutic action is medicinal action. They are pharmacologically desirable. That is called therapeutic action. So first we'll see a very important class of drugs called antacids. Antacids, we all know, are used in acidity, right? When excess acid is produced in the stomach, it leads to acidity, and for counteracting that, we take antacids, right? So a major breakthrough in the treatment of hyperacidity came through the discovery according to which a chemical, histamine, stimulates the secretion of pepsin and hydrochloric acid in the stomach. So there is a chemical which it stimulates the secretion of pepsin and hydrochloric acid. Now, when a person is suffering from acidity, we want to reduce the amount of hydrochloric acid. So somehow, if we could make sure that the chemical histamine is not produced or is inhibited, we could control acidity, right? So, the drug cimetidine or Tegamet, the structure is given in the figure, was designed to prevent the interaction of histamine with the receptors present in the stomach wall. So now histamine cannot send its signals to the receptors because of the presence of Tegamet, and hence hydrochloric acid production and pepsin production decreases, which leads to cure of hyperacidity. Now we come to antihistamines, which are another class of drugs. So histamine contracts the smooth muscles in the bronchi and gut and relaxes other muscles such as those in the walls of fine blood vessels. It is also responsible for nasal congestion. So there are synthetic drugs like brompheniramine, which is also called Dimetap, and terfenadine, also called Seldane. They act as antihistamines. So they reduce the amount of histamine. So they interfere with the natural action of histamine by competing with histamine for binding sites of the receptor. Right. So they do not let histamine attach to the binding sites because they compete. They go and attach themselves to the binding sites of the receptor so that histamine cannot continue with its natural action. Now, why do above mentioned histamines not affect the secretion of acid in the stomach? Right. The reason is that anti-allergic and antacid drugs work on different receptors. Right. This is very important. I told you that the drugs are very receptor specific. The binding sites are very shape specific, and a particular drug or a particular chemical will send its signals only to a particular kind of receptor. Right. Now, since the receptors on which the anti-allergic drug and the antacid drugs work are very different, the antihistamines do not really affect or alter the secretion of acid in the stomach, which is a good thing because we don't want it to happen. We want a particular amount of acid to be produced, and if a drug is performing certain action, it is always desirable that it should not hinder or it should not disturb the action of other receptors or other chemicals or important enzymes. Now, we discussed the third category of drugs, therapeutic drugs, called neurologically active drugs. So, they affect the nervous system in some way or the other. That's why they're called neurologically active drugs. So, the first among them are tranquilizers. So, tranquilizers are used for the treatment of stress and mild or even severe mental diseases. Now, there are various mechanisms by which tranquilizers function. Let's see how they function. Now, noradrenaline plays a role in mood changes. So, this is a chemical which affects mood changes, which causes mood changes, right? Now, if the level of noradrenaline is low for some reason, then the signal sending activity becomes low, causing depression. तो हमें कम नहीं होने देना है noradrenaline का level, वरना depression हो सकता है. तो in this case, antidepressant drugs are required, which inhibit the enzymes which catalyze the degradation of noradrenaline. So, what do antidepressant drugs do basically? वो जो enzymes noradrenaline के degradation को cause करते हैं, उन enzymes को catalyze कर देते हैं, उन enzymes का action को inhibit कर देता है. So, now the degradation of noradrenaline cannot take place. So, its level automatically increases, and which helps us avoid depression. So, this is how antidepressant drugs function. This is one mechanism by which tranquilizers can function. Now, if the enzyme is inhibited, if the enzyme which was catalyzing the degradation of noradrenaline is inhibited, उसका action कम हो गया है, तो noradrenaline अब धीरे धीरे बढ़ता जाएगा, metabolize होता जाएगा, and it can activate its receptors for longer periods of time. Now, this will obviously prevent depression. So, all these things are important because basically from board exams point of view, you need to remember the uses of drugs, the major examples, and how they function, the basics of all these things should be known to you. You can obviously avoid very complicated terms, but I guess these are the action by which the drugs function are very, very simple to understand. And just learning 2-3 major examples for each category of drugs should be enough. So yeah, examples of tranquilizers are iproniazid and phenelzine. Now, there are some tranquilizers like chlordiazepoxide and meprobamate are relatively mild tranquilizers. So they are mild, so they are used for relieving tension. Now, there is a tranquilizer called Equanilis, which is used to control depression and hypertension. Now, derivatives of barbituric acid, that is veronal, amityl, nebutyl, numenal and seconal, constitute an important class of tranquilizers. These derivatives are called barbiturates. Barbiturates are hypnotic, that is sleep-inducing agents. Some other substances such as tranquilizers are Valium and serotonin. So we saw so many examples, but out of them, barbiturates are important, Valium and serotonin are important. Their structures are also given. They do not need to be memorized, but yeah, you should know their names. And so you see how related all this is to our daily lives, right? We have acidity, we take antacids, then some people do take antidepressant drugs as well. So all these we see around ourselves each and every day. We see people talk about them, we see people buy and consume them, but we never really think about the chemistry behind these medicines because, well, we are too engrossed in solving, ionic equilibrium problems. But anyway, everything has its place. The next category of therapeutic drugs are analgesics. Analgesics reduce or abolish pain. Analgesics are basically painkillers, and well, I think they are the most often consumed class of drugs, right? Yeah, crocin, paracetamol are all analgesics, right? So they reduce pain, but they do not have any direct harmful effect like, impairment of consciousness, mental confusion, or incoordination or paralysis. All these are not there. So yeah, these can be classified as follows. One is non-narcotic or non-addictive analgesics. Examples are aspirin and paracetamol. Aspirin inhibits the synthesis of chemicals known as prostaglandins, which stimulate inflammation in the tissue and cause pain. So there are chemicals called prostaglandins which cause pain. They cause inflammation of the tissue. And what does aspirin do? It just stops these chemicals from being synthesized. It inhibits their formation, right? So these drugs are antipyretic. Antipyretic means they reduce fever and prevent platelet coagulation. Okay, they are used to prevent heart attacks also. So they're quite useful. And since they are non-addictive, it makes them even more desirable to be used. The second class is narcotic analgesics. So the examples like morphine and many of its homologs, they relieve pain and produce sleep. Tranquilizers are actually also used in sleeping pills. But yeah, anyway, narcotic analgesics are also called opiates because they were initially obtained from opium. Opium. These analgesics are chiefly used for the relief of post-operative pain, cardiac pain and pains of terminal cancer and in childbirth. So they are not taken casually. Because they are narcotic addictive.